Right. Let's go ahead and finish up our general virology section. And then we'll start talking about specific viruses. We'll jump into the papillomaviruses first. So kind of in this last section on viral diagnosis and treatment, one of the things that sometimes you can talk about with viruses, but really that only works if you are culturing them or infecting animals, is you can talk about the titer. So we've also used this term to talk about antibodies, like an antibody titer, basically the amount of antibody in the blood. So titer is also used to sort of quantify viruses. So it's the dilution that retains specific properties. So there's a tissue culture dose 50, causes effects in at least half of tissue culture cells. Lethal dose 50 that kills 50% of animals, and an infectious dose 50 that causes symptoms in 50% of animals. One of the other terms that we sometimes use is very similar to what we use for bacteria, where in bacteria we talk about colony forming units, but in viruses we talk about plaque forming units. So basically every time you have a zone of clearance that shows up in a cell culture, that's called a plaque, and it represents where one cell had been infected by a virus and died, and all the other cells nearby died too. Now, when you're doing culture or you're working with animals, just because you have a negative result doesn't mean that you don't have virus or that you haven't identified the right virus, just because these viruses are so difficult to work with. Again, it's one of those that I can't imagine that in most clinical lab settings that you would actually be working with the viruses themselves. Those of you who might be interested in like a microbiology research career, if you end up in labs that study viruses, you may be working with the actual viruses, but otherwise probably not. One of the other things that can make it difficult, especially to do things like calculate an infectious dose, is you could have animals that are infected and shedding viruses without showing symptoms. So it can make it challenging to determine how contagious the virus is, how many viral particles you need, and all of those things. So typically what we do to detect a viral infection, instead of trying to culture the organism, most commonly you're going to be looking for proteins or nucleic acids. And by proteins, you could be looking for two different things. Proteins produced by the virus, so you could be looking for viral antigens present in the host, or you could be looking for host proteins produced in response to the viral infection, namely antibodies. You would be looking for a host antibody response to the infection. So those are the most common ways that we diagnose viral infection. The second most common would be nucleic acid-based tests. Similar techniques to those that you would use for bacteria. So like with proteins, you would do things like Western blots, ELISAs for protein assays. For nucleic acids, really commonly you're going to be doing PCR or reverse transcriptase PCR if you have an RNA-based virus. You can do genome sequencing of the viruses because they're so small. It's pretty straightforward. So those are the most common. Culture is not terribly common. Again, just like with bacterial infection, if a patient has really high levels of IgM specific for a virus, it indicates an early infection. If you have that increase in IgG compared to IgM, that indicates a later infection. And we call it seroconversion when the antibody titer has gone up fourfold. So in an early infection, you may have X level of antibody. When it goes up 4 times that, or fourfold, that's when we call it seroconversion. But that's the same for anything. Limitations with serological tests are similar to those with bacteria. You can't necessarily tell when the infection began. You can have false positives and false negatives. With some of the viruses, it starts to get challenging when the viruses are closely related. One of the examples I'm thinking of is the flaviviruses, which is like West Nile, yellow fever, dengue. They all kind of are cross-reactive because they're all very, very similar viruses. And so you can get false positives. If you have one infection, it might tell you you have the other. So it can be a little bit challenging. Certain viruses like hepatitis B actually bind to antibody in serum and can complicate results. And again, cross-reactivity of antibodies, parainfluenza and mumps, for example, the yellow fever, West Nile, dengue complex. Sometimes the antibodies are so specific, if you're not testing for the exact right surface proteins, you're not going to get a positive result. So things like rhinoviruses tend to be very specific. And then in terms of treatment, again, the idea of treating a viral infection is challenging because a virus takes over the host cell. And if you're trying to stop a virus from replicating, what you're really doing is stopping the host cell from replicating. And if you're stopping host cells from replicating, you can kill host cells. So it can be really challenging to try to treat viral infections. But a lot of these different viruses have very specific things that they do, or very, very specific targets that we have antiviral drugs for. And as we talk about some of these specific viruses, we'll talk about how exactly those drugs work. But in this chart here, they give you examples. This is from the textbook, but they give you examples of different antivirals and what they can do to prevent viral infection or to help treat a viral infection. So again, as we talk about specific viruses, we'll talk about if there are therapies available and what those are. So to control viral infection, especially if you're working in a clinical setting, you want to do the best you can to limit personnel contact with sources of infection. So your personnel should always be wearing gloves. For respiratory viruses, possibly masks and or goggles. You'll quarantine for certain viruses. Improve hygiene, sanitation, and disinfection. If you have ever worked in a hospital, you most likely have had to demonstrate that you are immunized against a lot of very common viral infections. I know I had to, and if I couldn't prove it, they either tested me to prove it or they just gave me more vaccines. I didn't even work in the hospital. I just had to walk through the hospital sometimes, and they would do that. So, and then education. Okay. Again, envelope viruses are pretty sensitive to environmental conditions. So just by like cleaning the room with a disinfectant, you can usually get rid of most envelope viruses. Non-enveloped can be inactivated by things like ethanol, bleach. It requires just a little bit more in order to get rid of non-envelope viruses on surfaces. And again, as we talk about the different viruses, we'll talk about what you can do to control those infections. Okay, so let's jump in to some viral specifics. So the first viral group we'll talk about are the papillomaviruses. Papillomaviruses are naked. So again, when we think about the virus and we think about, is it naked or enveloped, that again has implications for how it can be treated, how it can be transmitted, right? So think about that. So it's a naked double-stranded DNA viral family, and the papillomaviruses are highly associated with transformation. So when we talk about transformation with eukaryotes, or because viruses infect eukaryotes, that's why we're talking about eukaryotes. Transformation in eukaryotes means becoming cancerous. Transformation in bacteria is picking up DNA from the environment. Same word, two different meanings depending on what organisms we're talking about. So if I tell you a virus is associated with transformation, I'm telling you it's associated with cancer, being a cancer-causing virus. It's oncogenic. With papillomaviruses, kind of the symptom you would see during an acute infection are warts. They can be cutaneous warts. They can be mucosal warts. And there are some strains of the human papillomavirus that are really highly associated with causing cervical, penile, anal cancers, but also oropharyngeal cancers as well. So they're more highly associated with causing cancers. There are over 100 types, but generally we split them into cutaneous versus mucosal. So mucosal are going to be the ones that can affect like the genital tract, the oropharyngeal regions, whereas cutaneous would be like a wart on your finger. But the thing is if you have a wart on your finger, it won't cause like genital warts as well. So cutaneous warts are cutaneous. So cutaneous strains are cutaneous. Mucosal strains are mucosal. So if you have a wart on your finger, you're not accidentally also going to give yourself genital warts. You could spread the wart on your finger to like, your arm or something if you're scratching, but it's not going to work that way. And it's the mucosal strains of HPV, human papillomavirus, that are more highly associated with causing cancer. When we talk about the viral genetics, I'm not going to expect you to remember like how big every genome is and how many proteins it makes and blah, blah, blah. That's not that important. I'm showing it to you so you can have it for your reference, but I'm not going to ask you on the exam, how many base pairs are in the human papillomavirus genome? But what I do want to point out is what happens to cause cancer with this virus. So normally what happens is you have, right, like 8 genes or so. And so in the viral genome, this protein E5 is what ends up first promoting growth of the cell. Because remember, in order for a virus to replicate, the cell machinery has to be on. The cell has to be replicating. And so what you'll end up having happen is you'll get a split in the genome, and we'll talk about that a little bit more detail in just a minute. But the genome, instead of being a circular genome, it will become linearized and integrate into the host chromosome. And when it does that, that's when you can get transformation to occur. So when you have a patient who has HPV infection, why are you beeping at me? There we go. So it infects the squamous epithelium of the skin and the mucous membranes. And so each type, either the cutaneous versus the mucosal, will go to one or the other. Okay. Now with the viral replication, I pointed out that gene E5 that has the epidermal, it stimulates growth, epidermal growth factor. So on your skin, right, we call that a wart. I'm sure everyone has either had a wart or seen a wart. You don't get them from kissing frogs. I don't recommend kissing frogs, but you're not going to get warts from it. You might get something else. I don't know. Parasites. But what you see, the cytopathic effect that you classically see with HPV infection are what are called coleocytes. And it's these cells here. It's the keratinocytes, but they have really shrunken, dense nuclei, and you can see that under the microscope. So that's the CPE that's visual through microscopy. And so what's happening is you have infection down here in these basal keratinocytes. The cells are proliferating. They're replicating the viral genome. The virus, right, eventually matures in some of these layers of tissue that are closer to the surface, and it releases, right, in the upper layers of the skin, if we're talking about a cutaneous form. And so it'll be shed, right, from these upper layers of tissue. So they are acquired by close contact. It's touching. Right? The tissue tropism, so is it cutaneous versus mucosal? It depends on the type, but it can persist. So if you've ever had a wart, if you've not had it removed, like medically removed, you might notice that it'll go away and come back, go away and come back, go away and come back, often in the same spot, because it's those cells down there that are infected with the virus. And so it will go latent for a little while and then it'll come back, go latent and come back. If you get it removed, like totally frozen or maybe like kind of, I don't know, scooped out or something, and they get all of the cells that have the virus, that's why you don't get the infection or you don't get the wart again. But if you just let it go away, or maybe you do like the little topical treatment, it may come back because you haven't gotten rid of that basal layer of cells that were infected. A wart is benign, right? It's not cancerous. It's not going to burrow into your body. Maybe it's unsightly. Maybe it's uncomfortable. Depends on where it is. Like if it's on the bottom of your foot or something, it probably hurts a lot more than if it's on like your forearm. But that's really it. It hides pretty well from the immune system, which is why if you have a wart, you probably don't experience like infection symptoms. It's not red, it's not swollen, it's just the overgrowth, right? But you don't have a big swelling area around the wart. There's no pus from immune cells that have rushed in there. It doesn't really cause an immune response. It's just an overgrowth of those epithelial cells. Eventually, they can resolve spontaneously. Your body does recognize when cells are doing the wrong thing, and they can be told to like tone it down. But there are certain types, especially mucosal types, that are associated with what we call dysplasia. The cells become different. And those can become cancerous. Now, is every patient who has ever had mucosal HPV going to develop cancer? Absolutely not. But of the types of cancer we see in those regions, anal cancer, penile cancer, cervical cancer, oropharyngeal cancer, are lots of those cancers associated with HPV? Yes. Okay. So HPV is not a cancer sentence, but a lot of people who have those types of cancers were infected with HPV. And you can detect it because the viral DNA is integrated into the host chromosome. So that's how we know that those patients have HPV. You can sequence the DNA of the tumor and find HPV in there. So the two strains of HPV that are most highly associated with cancer are two mucosal strains, HPV 16 and 18. Oh, I was wrong. I said 99.3. It's 99.7. Nearly all cervical cancer, 99.7% of cervical cancers have HPV DNA. And basically what that's trying to say is that, 0.3% of patients who get cervical cancer just got cervical cancer. It was unlucky that they developed cancer there. The rest of them, it was most likely because of HPV. And of those patients, that 99.7% who have cervical cancer with HPV, 70% of those patients have either 16 or 18. So that's why we say that those two are the most highly associated with causing cancer. So what happens, I told you they have a circular genome. So what happens to cause cancer is you get a break right here between what they're calling gene E1 and E2. When that happens, the virus actually can't replicate itself. But it can integrate into the host chromosome. So it's not making new viral particles. When it breaks and integrates, it's not making new viral particles. Instead, what's happening is, so these two genes that are responsible and helpful in making new viral particles don't work anymore because of where it's been broken. But the genes that still work are things like E5, which helps it respond to growth signals, and E6 and E7, which bind to P53 and P105. Maybe you remember from a class like cell and molecular, P53 is one of the most important tumor suppressor genes. So those viral proteins are still being made because the genome is integrated into the host chromosome. So now the host is transcribing and translating, and those proteins inactivate the tumor suppressor genes. So E5 is basically telling the cell, oh yeah, you should grow. Yeah, totally grow. And E6 and E7 are binding the stop signs that would have said, no, you shouldn't grow. So that's why you can get the promotion of cancer with this one. And so this is kind of what you're looking at. You have the infection in these, basal tissues, that basal layer, and this is going to be of the cervix. You have viral production, you have genital warts, okay? But at some point, you've gotten the integration of the viral DNA into the host cell, and the cells are no longer responding to growth signals and stop signals the way that they should. And so you can detect here where the cells will start to look different because they've started to not behave as they should. So you can sometimes see the changes in the cell as it's becoming cancerous, because cancer cells look different before it becomes invasive. But oftentimes it is not detected until it has become invasive. And so this would be considered cervical cancer as it's leaving the cervix. It metastasizes and goes to other areas. So that's the real dangerous thing about HPV. I imagine genital warts are uncomfortable and unsightly, but a genital wart isn't going to kill you. The real risk with HPV is and a finger wart's definitely not going to kill you. The real risk with HPV is the fact that it is so highly associated with all of those different kinds of cancers. Because it is naked, it's more resistant to being inactivated. So it can persist like in the dry skin for longer than you would think it should. And it can be transmitted on fomites, like surfaces or towels. Because again, it can persist. Infection can be acquired through breaks in the skin, sexual intercourse, or childbirth. Many patients will shed the virus without showing symptoms. So they could be shedding, but not have, active warts. I would imagine that most people, if they were going to engage in sexual activity and saw a bunch of warts, would be like, ooh, no. But you can still shed the virus without showing the warts. So it's estimated that this might be one of the most prevalent sexually transmitted infections in the US. The CDC estimates approximately 1/4 of the US population is infected with one or more strains of HPV, with 14 million new infections per year. But this is not something that is reportable to the CDC, so it's their estimate.
When you say that it can be transmitted on fomites such as surfaces and towels, would that be for both cutaneous and mucosal?
Yeah, both cutaneous and mucosal. Could be. So again, it is associated with other cancers, not just cervical. But of course, most cervical cancers are associated with HPV. About 70% of oropharyngeal cancers are associated with HPV. More than 60% of penile cancers, 90% of anal cancers, and 70% of vaginal and vulvar cancers. So like, for example, smoking and chewing tobacco also is associated with oropharyngeal cancers. But 70% have HPV as well. The most recent statistics are something like this. Again, because we tend to think of it as like a cervical cancer, we tend to think, oh, well, women get it the most. So women do get it the most, approximately 47,000 annual HPV-associated cancers in women, but there still are over 20,000 cases in men. In 2019, there were just over 4,000 cervical cancer deaths in the US, and it's estimated that approximately 10% of women that have one of the high-risk strains, so 16 or 18, will develop a cervical dysplasia. So they'll develop kind of that precancerous lesion. And when those are detected via pap smear, they can be removed. Risk factors would include multiple sexual partners. The more sexual contact you have, the more likely you might be exposed to different organisms. Smoking can put you at risk for developing cancer. Your genetics, immune suppression, so all of those things can put you at risk. Again, I'm not going to ask you to remember which types are more associated with what, except for the fact that types 16 and 18 are the most high risk for developing cervical cancer. That's really what I want you to remember. 16 and 18 are the most high risk. So, plantar warts, common warts, flat warts, all different kinds of warts. I didn't know there were multiple kinds of warts until I started taking classes like this. I'm like, wow, there's all different sorts of warts. But so, this person has a bunch of warts. So detection of cervical infection is generally done through a pap smear. Okay, it used to be that pap smears were recommended annually, especially for women who were sexually active. I feel like they changed it now to every 3 years or so. So, make sure that you put that on your calendar. Yes.
So there's no test for it like how they test for HIV or anything like that. It would only be able to be detected through a pap smear.
Mm-hmm. Yeah. So the thing with HIV that makes it easier to test for is HIV circulates through the bloodstream, so you just have to take a blood sample. To test for HPV, you'd have to do testing of the cervical sample, and then the fact that there's HPV in there may or may not be informative if the cells haven't turned, because again, they think 1/4 of people have it, and for most of those people, it's not going to be a big deal.
So technically, you can get a pap smear done, and you can have HPV, but it won't always be detected until the cells become irregular.
Mm-hmm. And even then, they don't always detect irregular cells. Sometimes they can miss it, and people can develop invasive cervical cancer. Either it develops really rapidly, maybe the pap smear wasn't done well, or the person who looked at it didn't do a great job. So
even if you get a pap smear,
you're
screwed.
Well, you have an option here, and I'll tell you your option. Well, okay, we'll get to your option in just a minute. Your warts can be removed. Recurrence is common unless you remove that basal layer. By stimulating the innate immune system, you can help kind of prevent the growth of cells and shedding of virus. Vaccines are available for the most common and most high-risk types of HPV. And I'm old enough to remember when this vaccine was first made available, and the FDA only approved it for preteen females. So at the time when it was first approved, it was only approved for females before the estimated age of onset of sexual activity. And there was a lot of controversy around this vaccine because HPV is thought of as a sexually transmitted infection. If you vaccinate people against a sexually transmitted infection, apparently it turns them into sex monsters, is what a lot of people were really worried about. Oh, they got a vaccine and now they're gonna go do things. And I'm like, well, no, they're probably already doing things, but if you give them a vaccine, you can protect them. Now it is recommended for both boys and girls. The recommendation is to give it before the onset of sexual activity. So I believe that the age that they'll first start doing the vaccination is 11 or 12 for the pediatric patients. But for breaking news, there are new findings that it can still be effective even in adults who have become sexually active and may have been exposed. It prevents cervical cancer to adults up to the age of 45. So it's now FDA approved for men and women up to the age of 45, because maybe you already have the virus, but the vaccine can help keep it under control and from developing cervical cancer. So if you have not been vaccinated against HPV, I imagine most of you probably have, because you are a lot younger than me and you probably were that age when you could have been vaccinated. But if you have not, CVS right there, at Gear in Monte Vista, they have it, because I saw it on the sign. I went and got my flu and COVID shot. They're like, HPV. And I'm like, oh, okay, maybe next time. I already got COVID, and I'm like, give me all the vaccines. I don't care. I'll take them all. So the vaccine can help.
So earlier you mentioned that infection could be acquired through childbirth. Childbirth.
Yeah,
so if they don't test for it until like you turn 21, how would you know if?
So generally, what you're going to have happen is it wouldn't be like a cervical infection that was acquired during childbirth.
Oh, okay.
So if anything, if maybe if the it could go maybe like oral, right, as the child is being delivered, but most likely not a cervical infection. So again, you would recommend the vaccination, because that can help even if the virus is there. Like when we talk about herpes viruses, we'll talk about chickenpox, and they'll still give. So like I've had chickenpox, but when I get older, they'll still give me the shingles vaccine to help prevent like the recurrence of it. So you could totally do something like that. Yeah. Okay. Other questions about papillomaviruses? Yes.
So it says that the FDA the vaccine up to like 45.
Mhm.
So do you know if they've done like any testing to see like for another vaccine for after, to protect you after you're 45, or they just don't care?
I think by the time you've hit 45, they just assume, they're like, well, you're probably sexually active, you probably already have it. They may expand it. I think it's probably just because they haven't had an opportunity to test the efficacy in patients who are older. I would think that's probably it. Because again, I think I've mentioned this before, we see a lot of sexually transmitted infections in elderly patients. So,
Interesting.
yeah, well,
They get a second wind.
they get their second wind, maybe they're widowed, or they've, once they're empty nesters, they decide, hey, we were together for the kids and not anymore, and now the kids are gone, so let's go do our thing. I don't know. But elderly patients. So if you work in geriatrics, good luck with you, But yeah, I think you'll end up seeing probably an expansion. There are a couple of different HPV vaccines. One only protects against a couple of strains, and then one is against like 9 different strains. So I think the more common one is called Gardasil.
Would most of them, or like would all of them protect against the major ones?
They all will, yeah. They'll all protect against the major, but then the Gardasil the one that's the 9 will protect against a few more of the other, like medium risk ones. Any other questions about HPV? So like I said, if you have health insurance and you've not been vaccinated against HPV, right there, the Walgreens or the CVS at Gear in Monte Vista, they had the little sign up. You can also get your COVID vaccine, by the way, and your flu. And they'll do them in the same arm, which I don't recommend. My arm swelled up like this big. I was like, what the heck? It looked like I had Lyme disease. I had like that bullseye-shaped like rash. My sister-in-law, what's wrong with your arm? I was like, oh, I got my COVID vaccine. Oh, I'm not going to get mine. I was like, no, get it. Just don't get the flu at the same time in the same arm. Don't do like I did. Get them in different arms. Okay. So adenoviruses are real common. I can almost guarantee that everybody in this room has had an adenovirus at least once, probably at least once this year. They're called adenoviruses because they were first isolated from an adenoid cell culture. So adenoids are like these little glands that you have if you haven't had them removed already. Sometimes they take them out at the same time if they take out your tonsils. They'll take out your adenoids if you end up like with a lot of infection in here. There are at least 100 serotypes About 52 of them can infect humans. Subtypes 1 through 7 are the most common. What we classically see in people most commonly are respiratory infections. This is one of the most common causes of the common cold. So that's why I say most of you, I would almost guarantee, have had an adenovirus probably within the past year. So I bet you've all had a cold in the past year. It also is a pretty common cause of pink eye, conjunctivitis, typically in children, not usually adults, but it's really, they're highly contagious. I'm sure my second grade teacher, when she saw one of the kids had pink eye, she was like, great, because everybody got pink eye. We all got it. But it can also cause more significant infection. It can cause some hemorrhagic cystitis, gastroenteritis. It can be fatal in some patients. But for the most part, in the majority of your patients, this is going to be a common cold. So you never will see them because most people don't go to the doctor when they have a cold. In other species, adenoviruses are known to be oncogenic. There are no known, I don't want to say it's not possible, but there are no known adenoviruses that infect humans that cause cancer. But in other species, some of these adenoviruses can be oncogenic. They are double-stranded DNA viruses. So we're gonna go with the DNA, the genetic material. So right now we're in DNA, so these are all gonna be DNA viruses for a while. Double-stranded DNA viruses, they are naked. This is what they look like under a microscope. And these fibers, they're proteins that stick out from the surface here are usually what they use to attach. So yeah, they have those fibers. They have their own DNA polymerase to help facilitate growth of the host cells and to escape the immune system. They can cause lytic, persistent, and latent infections in humans, most commonly a lytic infection. In some strains can immortalize animal cells, so they are oncogenic in other species. That's the important stuff to know about them. These are relatively quick. You know, it's one of those, somebody comes home with a cold and you're like, oh man, because you know within a day or two you're gonna get it too. The replication cycle is about 36 hours and one infected cell will release about 10,000 infected or infectious virions. So the burst size is about 10,000. They attach to glycoproteins on your cell surface, whereas a DNA virus, right, the nucleic acids are internalized into the nucleus. They can inhibit the immune response and promote the growth of the cell. So they don't want to be recognized and they want your cells to keep replicating. Because if your cell's replicating, that means they are replicating. Oops, did I go backwards? Oh, no, I did not. Okay, they're spread most commonly in aerosols, right? A cold, you're coughing and sneezing. They can be spread through fecal matter, the strains that cause gastroenteritis, and by close contact. Fingers spread the virus to the eyes. So like I said, they're naked. So fomite transmission is relatively common. So picture children, and then the other kid, like, right? They all are always touching stuff. Kids are gross. I have two of them, I know.